Hello again, students. Welcome back to Survey of Human Anatomy and Physiology, Biology 2404. We're going to continue looking at Chapter 15, the digestive system. This is Part 2. Before we move on, let me remind you that you should also be looking at digestive organs in your lab manual, doing your labeling and studying the anatomy of the digestive system. You should also be looking at the lab materials on the website with the pictures of the lab models and the videos that go with labeling the lab models, so that will help you with your lab practical over the parts of the digestive system. It also supplements our lectures so that you can better understand the anatomy part of the system. Let's continue by looking at the stomach. There's the cardia, the fundus, the body, and the pyloric region. And looking at these areas, this is the lower esophageal sphincter that leads into the stomach. This area is the cardiac region. This upper area is the fundic region of the stomach. This is the body of the stomach. The rogae is the ridges in the stomach, in the wall of the stomach. Cardiac in the area right after the esophageal sphincter. If the sphincter, by the way, a sphincter is a round, smooth muscle that opens and closes off, that would open and close off the stomach so that you don't have the backwash of the stomach contents into the esophagus. We just talked about that in part 1, and that would cause burning in that esophagus, often referred to as heartburn. The rogae is the gastric folds. Don't forget to look at the models so you can identify these parts on the model as well. This is the pyloric region of the stomach, and then the pyloric canal. It leads to the pyloric sphincter, and what did I say a sphincter was? Smooth muscle, and a round smooth muscle that closes off. A sphincter muscle will cause opening and closing or dilation, constriction or dilation of this opening. Constriction would be closing it off, and dilation opening it up. So we've seen sphincter muscles before, and this would be the pyloric sphincter leading into the duodenum. Okay, so know these parts of the stomach. The rogae with gastric folds have mucosa. You have mucosa all through this, and submucosal layers, and with those submucosal layers adding mucus. We'll look at that in just a minute. The valve, the pyloric sphincter, you had two valves mentioned, the lower esophageal sphincter and then the pyloric sphincter. Smooth muscles that can constrict or dilate to allow substances to move through. Again, you have two layers of muscles here. This longitudinal layer, this outermost layer, is a longitudinal layer, and then you have a layer that goes this way around the circumference of the stomach, and they alternate in contraction and relaxation to give you that churning of the contents. The gastric glands secrete gastric juice. One of them is pepsin. pepsin is an enzyme that begins the breakdown of proteins. So we talked about the beginning of the breakdown of carbohydrates in the mouth with saliva and amylase. Pepsin aids in the breakdown of proteins, and remember, proteins are broken down into polypeptides. Now, what is a peptide? A peptide is the bond between two amino acids. So we call a string of amino acid peptides, polypeptides, many amino acids, down to a dipeptide. Di is two amino acids all the way to the individual amino acids. So we have to have our proteins broken down into amino acids so that we can use those amino acids to build more proteins and other substances that we need. So pepsin is in the gastric juice. It's an enzyme that helps to break down proteins. It is produced by what are called chief cells in the lining of the stomach. You have hydrochloric acid. There are types of cells called parietal cells that produce hydrochloric acid, an intrinsic factor, and hydrochloric acid helps to create the specific pH needed for the pepsin to work. If you don't have that acidic condition or environment in the stomach, then the pepsin will not work. So hydrochloric acid is produced so that you can have a very acidic environment for pepsin to work. Intrinsic factor helps you absorb vitamin B12. The hydrochloric acid intrinsic factor, again, produced by parietal cells. You have mucus cells that produce mucus that helps to protect the stomach lining, so that, by the way, mucus is more alkaline, so it is produced by these mucus cells, so the lining of the stomach is protected from the gastric juices, the pepsin and hydrochloric acid, because it would break down and cause an irritation on the wall of the stomach. So the gastric glands produce the gastric juices, and the chief cells in the gastric glands produce the pepsin, and the parietal cells produce the hydrochloric acid and the intrinsic factor. Pepsinogen, PEPSINOGEN, is a substance that activates the pepsin. Gastrin, here's another one of those hormones, is released. It is a hormone that causes the release of gastric juices. Okay, gastrin is a hormone that causes the release of gastric juices. And by the way, just the smell of food can activate that hormone, that gastrin, that will signal the release of the gastric juice. So that's why sometimes when you're hungry and you smell something good, then your stomach will start growling, because it's getting ready to receive that food. There's stretch receptors that help the churning of that food. The chyme is the food and the gastric juice mixed together. It holds the stomach holds approximately 1 liter of fluid, or chyme, and very little absorption occurs in the stomach. It's a processing place. So you have some salts, water, and alcohol is absorbed in the stomach. Much everything else is absorbed in the small intestines. We have the continuation of the breakdown of carbohydrates. If it didn't completely happen in the mouth, it continues in the stomach, and it finishes off in the stomach into monosaccharides very quickly. It takes a little bit longer to break down the proteins in the stomach with pepsin. And if it is a fatty food, it takes even longer, maybe 3 to 6 hours to break down the fatty foods. And that is usually continued on in the small intestines, but it will release the carbohydrates or the monosaccharides very quickly. The proteins broken down into amino acids soon after that, and then the fatty foods 3 to 6 hours before they're broken down. Now, some certain lipid-soluble drugs are absorbed through the stomach, along with those salts, water, and alcohol. The chyme is moved through the pyloric sphincter to the duodenum, where the pancreas, liver, and gallbladder add their secretions. The pancreas extends horizontally across the abdominal cavity here, and the head is in a C shape in this area. Curve of the duodenum. Most of the cells secrete digestive enzymes into the ducts, which empty into the duodenum. So you have the minor and major duodenal papilla, and these ducts. Here is the gallbladder. Now, bile is sort of a green color, so you'll usually see, and the gallbladder is sort of a green color. So on the models, you'll see the gallbladder in green, and the bile duct, and the left hepatic duct and common hepatic duct, and the right hepatic duct coming from the liver. The liver produces the bile that's stored in the gallbladder, and it comes down into the bile duct, mixes with the pancreatic substances into the duodenum. Digestive enzyme secreted by the pancreas is dumped into the duodenum from the pancreas in this area, in this C-shaped curve of the duodenum. And you have the hepatopancreatic sphincter that regulates the dumping of the bile and the digestive enzymes from the pancreas into the duodenum. Sodium bicarbonate is secreted by the pancreas, and what it does is neutralize that acidic chyme. It's very acidic, and then the sodium bicarbonate, when we have acid indigestion, what do we take? Sodium bicarbonate. And the pancreas produces this to help to neutralize that very acidic chyme. It's released in response to the hormone secretin. Other pancreatic enzymes. There is an enzyme for every food group. Amylase is released in the mouth and started in a little bit by pancreas too, to continue the breakdown of those carbohydrates into simpler sugars. Lipase, this ASE ending indicates that it is an enzyme. Anytime you see that ending, just think of enzyme. We already saw that a lipid is a fat, lipase to break down fats, and protease to break down proteins, and nuclease to break down nucleic acids. These are produced by the pancreas. Okay, the amylase to further break down the carbohydrates, lipase to break down fats, protease to break down protein, along with chymotrypsin and trypsin and carboxypeptidase are different amino acids that are produced by the pancreas to break down proteins. These are secreted into the duodenum of the small intestines. It's released in the response to the hormone cholecystokinin is released to increase gastric enzymes, and less cholecystokinin means less of the enzymes released. More cholecystokinin, more it increases gastric enzyme excretion. And as you will recall from the endocrine chapter, the pancreas also is stimulated to produce insulin with high blood sugar, and then glucagon when there is low blood sugar. And let's see how that works with the liver. A single liver cell can carry on over 500 metabolic events at any time. It's involved in carbohydrate, lipid, and protein metabolism. It's divided into lobes. You have this smaller left lobe with the round ligament here, and then the very large right lobe, and then the lobes are divided into smaller lobules. These lobules are the functional units of the liver. You have oxygenated blood from the hepatic artery, and the blood high in food from the hepatic vein mixes freely in the sinusoids, this area. And these are channels that have nutrients that nourish the liver cells. Also, you have the with high sugar content in the blood or high glucose content in the blood, you would have the release of insulin, and so the response of the liver is to pull that glucose out of the blood and store it in the liver as glycogen, a more complex form molecule that can later be broken down into glucose again and used. And then that's with insulin. And then if you have a low glucose level in the blood, then with the hormone glycogen that was produced by the pancreas, the liver can, in this area, help to break down the glycogen into glucose to increase the sugar content, the glucose content of the blood. So the hormones have an effect here on what's happening in the liver, plays a role in what's happening in the liver. Okay, so remember the insulin with high blood sugar, the liver will pull that the glucose out of the blood and store it in the liver, that the hepatic cells, as glycogen. And then when the blood sugar drops and you have glucagon, the liver will be stimulated into breaking down that glycogen into glucose, so it will enter the bloodstream and raise the blood sugar levels. Okay, so then you have the liver decides what to do with the nutrients. The copper cells clean the blood. They're large phagocytic cells, and phagocytic cells or macrophages are like Pac-Man. They remove bacteria and other foreign particles that are in the blood that could be a threat. They have digestive enzymes inside of them to break these down, and we're recycling machines, so whatever we can use we'll keep and get rid of whatever's left over. But that's the job of the copper cells. The gallbladder's function is to secrete or actually is to store the bile made by the liver. It concentrates and stores the bile made by the liver. It is a yellowish green color liquid. The bile is released in response to cholecystokinin. It consists of bile salts, water, bile pigments, bilirubin and biliverdin, which gives it its color, cholesterol and electrolytes. The only part that play a role in digestion is with the bile salts. It combines with lipids and fat-soluble vitamins and aids in their absorption through the intestinal wall. It helps to emulsify fats. Now, what does emulsify mean? Well, okay, so when you're washing your dishes, you know that even if you use very hot water, it doesn't do a very good job on breaking down the food particles or the fats in the food particles on the dishes unless you have some detergent. Detergent is an emulsifier. It helps to break down the fats, and that's the action that is played by emulsifiers in the liver. In the gallbladder, the function of the bile is to emulsify the fat, so lipase has more surface area to act upon. So that dish detergent that you use in the soapy water, it helps to break down the fat into smaller globules, and that's what happens here. It helps to also neutralize acidic chyme. It emulsifies fats and also makes the fats more water soluble. The small intestines, it receives secretions from the pancreas and the liver, and here you have the completion of the digestion of the nutrients. It absorbs the products of digestion. It transports the residues to the large intestines. Okay, the duodenum, we already talked about the duodenum, is this sort of C shape coming off the stomach. And then you have the jejunum, leads to the ileum. Okay, leads to further on down here is the ileum. And to the ileocecal valve that leads into the cecum. You have mesentery here with these blood vessels. This area is very vascular because, again, the small intestines have the function of completing digestion of nutrients and then absorbing the products of digestion. So all the substances that are needed by your body, those amino acids, the glucose amino acids, the nucleic acids, the glycogen and fatty acids are all absorbed here through the walls into the blood supply and taken to the tissues that make use of these nutrients. That small intestines is suspended by the mesentery. And you see the different parts here. Okay, the stomach, the duodenum, to the jejunum, leading to the ileum, to the ileocecal valve, another one of those sphincter muscles surrounding that valve, allowing the product, basically by the time you get to the cecum, you have the products that are left over after all the nutrients have been absorbed that need to be absorbed. You're going to have water and waste products that are dumped into this mesentery or into this cecum. Okay, here's the appendix. The small intestine's wall, we have the villi, those finger-like projections, and then on the ends of these you can have the microvilli. So if you were to take a string and line it up on this surface and then stretch that string out, you would find that it has a very large surface area, and that's the purpose of this, the villi, is to, and the microvilli. Stretch all this out and you're going to have a lot of surface area, so it increases surface area. The placae circularis are the folds, or plicae are the folds. And then you have the submucosa here, the muscular layer, and the circular layer going around the circumference, and the longitudinal layer, and then the serosa. And remember, the serosa is the visceral peritoneal layer. You have simple columnar epithelium, that columnar or column shape. Now remember, we talked about simple before. That means a single layer of rectangular epithelial cells. Okay, when it's a single layer, it allows for the passing of materials through that, the nutrients that need to be passed on to the blood. And here you have the blood vessels. You have an arteriole and a venule and capillaries here. And then you have a lymph vessel and the lacteal with the blood capillary network, because all those nutrients need to be picked up by the blood and taken to where they're needed. Okay, remember, the lumen is the opening in that tube, the small intestines, and there's the villus. A villus is one, villi is many of those finger-like projections. And then you'll have goblet cells there, and remember, goblet cells for mucus, helps things to move along. Then on the surface of the villi, you're going to have microvilli. So you have those finger-like projections in that intestinal wall, but then on the surface you're going to have microvilli that increases the surface area even more. So this is the surface layer of that columnar cell, and here's the lumen. So the surface layer is called the apical layer. What's exposed to this lumen or open surface are microvilli. So they're like mini villi, that microvilli, on the surface of that cell. And the purpose, increased surface area for more absorption of what's needed to be passed on to the blood supply. Mucus is given off by those goblet cells. Intestinal juice with no enzyme that neutralizes the acidic chyme. Mucosal cell enzymes will be released that are embedded in the microvilli. Final breakdown of food as it is absorbed. Peptidase helps to break down that polypeptide, the dipeptide, into amino acid. Sucrase helps to break down and maltase and lactase, broken down into glucose and fructose. Lipase for the completion of the breakdown of the fats. Remember, the sucrase, maltase, and lactase are the enzymes that help to break down those disaccharides, those sugars, into usable parts, the glucose and the fructose. Lipase, the fats, help finish the breaking down of those fats. And enterokinase and trypsinase, trypsin, help continue breaking down of the proteins, help to break down the protein. You have absorption, the movement through the body wall into the bloodstream, and what moves through the final products. And these can be used by the cellular machinery, monosaccharides, glucose and fructose, your nucleic acids, the amino acids, electrolytes, and water. They enter the capillary networks of the blood vessels to the blood. Fatty acids are absorbed into the lymph. Now the large intestines. Remember that you have the sphincter here that leads into the ileocecum sphincter that leads into the cecum, the beginning area, and then you have the appendix that comes off of the cecum. Ileocecal sphincter regulates it. It dilates or constricts to allow for the movement of the substances into the cecum. The ascending colon, so the food is going to move ascending, moving up. Transverse colon, descending colon. And then when you have this S here, think of the S, is the sigmoid colon, then the rectum, and the anal canal to the anus. You have muscular layers here. You have the sphenic flexure, these grooves here that help to move things along. Mucus to add moisture. You have absorption of water here, and some lipid-soluble vitamins are about the only things that will pass through here. You needed a lot of fluids for digestion, and this helps to return water to the bloodstream, and that's why you need to drink a lot of water, or enough water, to keep that substance that's traveling through the digestive system to be very liquid. You need it to be very liquid. Here are those layers again: the serosa, the muscular layer, and the submucosa, the mucosa, and there's the lumen. With the goblet cells, you have that mucus being added all the time. There's little or no digestive function. It absorbs water and electrolytes, maintain that sodium, potassium, and the other electrolytes. It secretes mucus. Mucus helps that waste material move along. It's that water is being reabsorbed and sent to the bloodstream, so you need something to help move the rest of the waste materials along, so that mucus helps that. It houses the flora and fauna, the intestinal flora. It forms feces and is carried out by defecation. And that is the end of the chapter on digestion. Don't forget to visit the lab book, lab manual. Fill out the digestion lab exercises, lab exercise 39, and review the models on the website with little videos that go with the models that you'll be responsible for.